In our last video, we explored two key experiments. First, we tested whether these bacteria called lactic acid bacteria or LAB can survive in the presence of pesticides. Then, we saw if they could actually bind to the pesticide molecules themselves and remove them from the environment. If you missed that video, I highly recommend watching it first so you get the full picture of how these tiny bacteria are working to protect honeybees. Link in the description. Today, I want to show you two more key experiments from the same study. Can dead bacteria still bind pesticides? And can these bacteria protect real living cells, including insects and human cells, from pesticide toxicity? Let's get into the details. In this experiment, the researchers wanted to know if LAB could still bind pesticides even after being heat inactivated, which means the bacteria are no longer alive. Why? Because this way scientists can determine if the phenomenon is purely physical-chemical or if it depends on the bacteria's active living metabolism. It is an important information in case we want to try to use this information to fight pesticide exposure around us. So they took the same lactic acid bacteria strains from the previous experiments, but this time they killed the bacteria by heating them up. The answer for the experiment? No. Dead bacteria were unable to bind to pesticides at all. Only live, active bacteria were able to effectively capture and detoxify the chemicals from the environment. Very interesting. In this next experiment, the researchers wanted to take things one step further and see if LAB could actually protect living cells from the damaging effects of pesticides. They tested 3 types of cells, one from insects, one from rat intestines, and one from human intestines, by exposing them with pesticides with and without lactic acid bacteria present. And the results were pretty clear. The cells that had LAB bacteria present experienced significantly less damage from the pesticides compared to those without. This suggests that LAB might not only help protect honeybees, but could also have broader implications for protecting other organisms, including us. As exciting as these results are, it is important to point out that this specific study only looked at in vitro experiments, meaning they were only done in the laboratory using isolated cells. There is still no in vivo testing with live bees at this point as far as I know, which will be the next step in understanding how these bacteria work in a real hive environment. I'm going to keep an eye out for research involving live bees, and as soon as I find any updates, I'll be sure to bring that information to you. Please sign up to my email list to be notified earlier. Link in the description. Also, just like I mentioned in the last video, no single scientific article gives us the full story. It is crucial to remember that only time and more research can offer a more complete understanding of these findings. Science is an ongoing process, and it is up to all of us to stay curious and look for potential flaws or gaps in experiments. So if you have thoughts or suggestions for further research, leave them in the comments below. I'd love to hear them. A big thank you to Strong Microbials for sponsoring this video. You can actually try out their microbials products with your own bees and run your own experiments. The link, it is in the description. So give it a shot and see if you notice any improvement on your bees or not. And don't forget to come back here and let me know your results. Deal? Great. Stay tuned for more and don't forget to subscribe and hit the bell button. Thanks for watching. Inside the Hive.tv, the show about bees. See you guys in the next video.